Let's begin by understanding something fundamental. Eating is not just about fueling the body, it's about behavior, biology, and neurochemistry. When we talk about healthy eating, most people think of macronutrients, carbs, proteins, fats, and micronutrients, vitamins, and minerals. But what's often missed is how eating behaviors are regulated by brain circuits, specifically circuits that govern reward, stress, and decision making. There are systems in the brain, like the hypothalamus, that track energy needs. But there's also the mesolimbic dopamine system, which responds to the anticipation and pleasure of eating, especially hyper-palatable foods. That means what you eat is influenced both by what your body needs and by how your brain interprets reward, habit, and even emotion. Now let's talk about eating disorders, which are not simply about willpower or lack of discipline. These are complex neuropsychiatric conditions. Eating behaviors are regulated by complex brain circuits that involve multiple interacting systems, including those that process reward, stress, and decision-making. At the most fundamental level, eating is necessary for survival, and the brain is hardwired to ensure that the body gets enough energy to function. The hypothalamus plays a central role by monitoring energy needs through hormones such as leptin, which signals satiety, and ghrelin, which signals hunger. However, the hypothalamus doesn't work alone. It is in constant communication with other areas of the brain, such as the limbic system and the prefrontal cortex, to integrate emotional, environmental, and cognitive inputs. These connections mean that what we eat and how much we eat is not simply a response to physical hunger, but also to cues from our surroundings, our emotional state, and learned habits. The brain's reward system, particularly the mesolimbic dopamine pathway, plays a critical role in eating behavior. When we anticipate or consume food, dopamine is released in the nucleus accumbens, creating a sensation of pleasure or motivation. This dopamine release is not necessarily proportional to the nutritional value of the food. Highly processed, calorie-dense foods that are high in sugar, fat, or salt can cause a stronger dopamine response than nutrient-rich but less palatable foods. Over time, repeated exposure to these hyper-palatable foods can alter the brain's reward threshold, meaning more stimulation is needed to achieve the same sense of satisfaction. This mechanism is similar to how the brain adapts in substance addiction, leading to cravings and compulsive eating behaviors. Stress also exerts a powerful influence on eating behavior. When the body experiences stress, the hypothalamic pituitary adrenal HPA axis activates, releasing cortisol. Cortisol can influence appetite and food preference, often increasing the desire for energy-dense foods that are quick sources of fuel. This is thought to be an evolutionary mechanism preparing the body for fight or flight by ensuring adequate energy availability. However, in modern contexts where physical activity doesn't always follow stress, this can lead to patterns of emotional or stress-induced eating. Chronic stress can disrupt normal hunger and satiety cues, making it harder to maintain balanced eating behaviors. The prefrontal cortex, which is involved in decision-making and impulse control, is another crucial player. This region helps regulate the more primitive drives of the limbic system by applying rational thought, planning, and long-term goal setting. In the context of eating, the prefrontal cortex allows individuals to choose foods that align with health goals rather than immediate cravings. However, when the prefrontal cortex is compromised by fatigue, emotional strain, or environmental temptations, its regulatory capacity weakens. This makes it more likely that decisions will be driven by the immediate reward of taste and pleasure rather than nutritional value. Environmental cues also play a significant role in activating these brain circuits. Visual and olfactory stimuli, social situations, and even advertising can trigger the reward system, leading to cravings even in the absence of physiological hunger. This happens because the brain has learned to associate certain cues with the rewarding experience of eating. Over time, these associations become conditioned responses. Further reinforcing the behavior, it's important to note that these circuits are bidirectional. Eating influences the brain, just as the brain influences eating. Nutrient intake affects neurotransmitter production, hormonal balance, and energy availability, which in turn affect mood, cognition, and motivation. For example, amino acids from protein are necessary for synthesizing dopamine and serotonin, while glucose is a critical energy source for brain function. Deficiencies or imbalances in these nutrients can alter mood and decision-making, creating feedback loops that influence future eating behaviors. These systems are highly adaptable, which is both a strength and a vulnerability. The adaptability allows for survival in varying environments, but it also means that modern food environments, filled with easily accessible, highly rewarding foods, can hijack these systems. In such environments, Eating behavior can become dysregulated, with the reward system favoring immediate gratification over long-term health. This is why understanding the interplay between reward, stress, and decision-making systems is essential for promoting healthier eating patterns and preventing disordered eating behaviors. Anorexia nervosa is a complex condition characterized by severe food restriction, an intense fear of gaining weight, and a distorted perception of body image, and its roots lie in both psychological and neurobiological mechanisms. The brain plays a significant role in how anorexia develops and persists, with certain areas showing altered activity and communication patterns that contribute to the symptoms. One key region involved is the insula, which is responsible for interoception, the ability to perceive internal bodily states such as hunger, fullness, and physical comfort. In individuals with anorexia, the insula often functions differently, leading to a misinterpretation of internal signals. They may perceive themselves as full after consuming very little food or may not register hunger in the same way that others do. This misalignment between physiological needs and perception plays a role in the ongoing restriction of food intake. The prefrontal cortex, particularly areas involved in cognitive control and inhibition, is also more active in anorexia. This heightened activity can allow individuals to suppress natural hunger cues for extended periods of time, maintaining restrictive eating patterns despite strong biological signals to eat. In a sense, the prefrontal cortex overrides the body's natural survival instincts, enabling the person to adhere to rigid food rules and avoid situations that involve eating. While cognitive control is normally beneficial in helping with decision-making and planning in anorexia, it can become maladaptive, contributing to obsessive thoughts about weight, shape, and food. Reward processing in anorexia also shows distinct differences. Normally, the consumption of food activates the brain's reward system, releasing dopamine and creating a feeling of pleasure and satisfaction. In individuals with anorexia, this dopamine response to food is blunted. This means that eating may not feel rewarding, even when the body is in a state of energy deficit. Instead of experiencing pleasure from eating, individuals may feel anxiety or discomfort, which further reinforces restrictive behavior. Over time, this lack of reward from eating can become a self-perpetuating cycle, as the brain learns to associate eating with negative emotional states rather than positive ones. Additionally, alterations in the serotonin system have been observed in anorexia. Serotonin plays a key role in mood regulation, appetite, and impulse control. Elevated serotonin activity in certain brain areas may contribute to the heightened anxiety and obsessive compulsive tendencies often seen in anorexia. Restricting food intake can temporarily lower serotonin levels, which might paradoxically reduce anxiety for some individuals, reinforcing the drive to continue restrictive behaviors. This biochemical loop can make recovery challenging, as refeeding and normalizing eating patterns may initially increase anxiety until the brain adapts. The amygdala, a region involved in emotional processing and threat detection, is also implicated in anorexia. Individuals with anorexia may experience heightened amygdala activity when confronted with food-related stimuli or situations that challenge their restrictive behaviors. This can create intense emotional reactions to eating, body image discussions, or even the idea of weight restoration. These reactions are not just emotional in nature, but are tied to physiological stress responses, with increased cortisol levels and activation of the hypothalamic pituitary adrenal axis. Body image distortion in anorexia is partly linked to altered neural processing in visual and parietal regions of the brain. These areas help integrate sensory information and create a mental representation of the body. In anorexia, the way the brain processes visual and sensory input may lead to a persistent perception of being overweight, even when the body is significantly underweight. This distortion is resistant to correction through logic or external feedback because it is embedded in the way the brain interprets sensory information. Anorexia is also associated with changes in neural connectivity between brain regions. Communication between the prefrontal cortex, insula, and limbic system may be altered, creating a network that reinforces restrictive behaviors and rigid thinking patterns. The prefrontal cortex may exert excessive control over the limbic system, suppressing emotional responses to food and overriding instinctual drives to eat. At the same time, disrupted connectivity may impair the ability to adapt behaviors or shift away from entrenched thought patterns, making recovery more difficult. Genetic and environmental factors interact with these neural mechanisms. Certain genetic predispositions may make individuals more susceptible to heightened anxiety, perfectionism, or altered reward processing. Environmental factors such as societal pressure, stressful life events, or early childhood experiences can trigger the onset of anorexia in individuals who are biologically vulnerable. Once the disorder develops, The altered brain circuits reinforce the behaviors and thoughts that maintain it, creating a feedback loop that becomes increasingly resistant to change. The persistence of anorexia despite significant health risk highlights how deeply ingrained these neural and psychological patterns can become. The brain's adaptability, or neuroplasticity, can work against recovery by strengthening the circuits that support restrictive behaviors. However, the same plasticity also offers hope, as targeted interventions can help rewire these circuits over time. Understanding anorexia as a disorder involving specific brain mechanisms helps move away from oversimplified ideas about willpower or choice, and instead frames it as a condition requiring approaches that address both the mind and the underlying neurobiology. Bulimia nervosa is characterized by repeated cycles of binge eating followed by compensatory behaviors such as self-induced vomiting, excessive exercise, or misuse of laxatives, and its underlying mechanisms are deeply connected to neurobiological and psychological processes. At the core of bulimia is a dysregulation of the brain's reward system, particularly in circuits involving dopamine and serotonin. Dopamine, a neurotransmitter associated with motivation and reward, is highly active in anticipation of binge eating, creating an intense drive to consume large amounts of food in a short period. Unlike typical eating episodes that are guided by hunger and satiety, the binge episodes are often triggered by emotional states, stress, or exposure to highly palatable foods, with the dopamine system amplifying the urge. The serotonin system, which plays a significant role in mood regulation, impulse control, and appetite, also shows altered functioning in bulimia. Low serotonin activity is associated with increased impulsivity, mood instability, and difficulty regulating emotional responses. This can make it harder to resist urges to binge, particularly when experiencing negative emotions or stress. After a binge episode, serotonin levels may temporarily rise, which can bring brief relief from emotional distress, but this is quickly followed by guilt, shame, and anxiety. These negative feelings can drive the compensatory behaviors that define the disorder, such as purging, as a way to relieve both physical discomfort and psychological distress. The prefrontal cortex, which is responsible for decision-making, self-control, and long-term planning, often shows impaired functioning in bulimia. When the prefrontal cortex is less able to regulate impulses, the more primitive drives of the limbic system, particularly the nucleus accumbens, dominate behavior. The nucleus accumbens responds strongly to cues associated with rewarding experiences like food, making it more likely that a person will engage in binge eating despite intentions to avoid it. This reduced top-down control from the prefrontal cortex means that decisions are often driven by immediate emotional states and cravings rather than longer-term health considerations. Stress plays a significant role in the cycle of bulimia. When the hypothalamic pituitary adrenal HPA axis is activated under stress, cortisol is released, which can influence appetite and food-seeking behavior. For some individuals, elevated cortisol levels enhance cravings for calorie-dense, high-sugar foods that provide quick energy. These foods also activate the reward system more strongly, which can intensify binge episodes. Following a binge, purging behaviors can be driven by both emotional and physiological states. Active. Purging can temporarily relieve feelings of fullness and reduce anxiety by creating a sense of regaining control, even though it does not effectively eliminate all consumed calories and has harmful effects on the body. Emotional regulation difficulties are another central feature of bulimia. Many individuals use binging and purging as maladaptive coping mechanisms to manage overwhelming emotions, stress, or feelings of emptiness. This is tied to altered connectivity between the amygdala, which processes emotional responses, and the prefrontal cortex, which should regulate them. When this connection is weakened, emotional distress can more easily trigger maladaptive behaviors, and rational strategies for managing emotions are less accessible in moments of stress. Repetitive cycles of binging and purging also create changes in the brain and body that reinforce the disorder. Repeated binge episodes can alter insulin and leptin signaling, disrupting normal hunger and satiety cues. Purging, especially through vomiting, can lead to electrolyte imbalances and affect the body's physiological stability, which in turn can influence mood and cognitive function. Over time, these physiological changes interact with brain circuitry, making it harder to break the cycle of disordered eating. Environmental cues can further perpetuate bulimia. Food cues, stress-inducing situations, or even certain social environments can trigger activation of the reward system and create strong cravings. This is due to learned associations between these cues and the rewarding aspects of binge eating. Over time, the brain becomes conditioned to respond to specific triggers with binge behaviors, similar to the way substance use can be triggered by environmental reminders. Genetic and personality factors can also predispose individuals to bulimia. Traits such as impulsivity, high sensitivity to reward, and emotional instability can increase vulnerability. Combined with environmental stressors, these traits can contribute to the onset and maintenance of the disorder. While bulimia may appear to be a behavioral choice from the outside, the underlying neurobiology shows that it is driven by powerful, dysregulated brain circuits that are not easily controlled through willpower alone. The bulimia's cycle of binging and purging can persist because each phase temporarily alleviates the distress caused by the other. The binge temporarily numbs or distracts from emotional pain, and the purge temporarily reduces guilt and physical discomfort. However, both behaviors feed back into the same brain systems that create cravings, impulsivity, and emotional instability, making the disorder self-perpetuating over time. Understanding these underlying mechanisms helps reveal why bulimia is resistant to simple interventions and why targeted approaches that address both brain function and emotional regulation are necessary for recovery. Binge eating disorder is characterized by recurrent episodes of consuming unusually large amounts of food in a relatively short period, accompanied by a feeling of loss of control. And unlike bulimia, these episodes are not followed by purging behaviors. At the core of this disorder lies a dysregulation of the brain's reward and emotional regulation systems. The reward circuitry, particularly involving the mesolimbic dopamine pathway, is highly responsive to food cues, especially calorie-dense, highly palatable foods that combine sugar, fat, and salt. When individuals with binge eating disorder are exposed to these cues, dopamine release in the nucleus accumbens creates a strong motivational drive to seek and consume these foods, often independent of physical hunger. Over time, repeated binge episodes can lead to adaptations in this pathway, including a reduction in the sensitivity of dopamine D2 receptors. This means that the brain requires more food to achieve the same level of reward, creating a cycle similar to that seen in addictive behaviors. Emotional triggers are a significant driver of binge eating episodes. Many individuals with binge eating disorder experience heightened emotional reactivity and difficulty regulating negative emotions. The amygdala, which processes emotional responses, can become overactive, while its communication with the prefrontal cortex, which is responsible for regulating impulses and planning, may be weakened. This imbalance means that in moments of stress, anxiety, sadness, or boredom, the regulatory influence of the prefrontal cortex is diminished, making it more likely that the individual will turn to food as a coping mechanism. The act of eating can temporarily soothe emotional distress by activating the brain's reward system and modulating stress-related neurochemicals, but this relief is short-lived, often followed by guilt or shame, which can further perpetuate the cycle. Unlike typical overeating, binge episodes are not simply the result of poor dietary choices or occasional indulgence. They involve a compulsive quality, where eating continues past the point of physical comfort and is accompanied by a sense of being unable to stop. This loss of control is linked to the interplay of multiple brain systems. The insula, which processes interoceptive awareness, may function differently, leading to impaired perception of fullness or satiety during binge episodes. At the same time, the orbitofrontal cortex, which evaluates the reward value of food, can remain highly engaged, continuing to signal the rewarding aspects of eating even as discomfort sets in. Physiological factors also contribute to the maintenance of the disorder. Binge eating often disrupts normal appetite, regulating hormones such as leptin and ghrelin. Leptin, which signals fullness, may become less effective, while ghrelin, which stimulates appetite, can remain elevated or dysregulated. This hormonal imbalance, combined with altered dopamine signaling, creates an environment where cravings are frequent and strong and satiety signals are muted. Repeated binge episodes can also lead to fluctuations in blood sugar levels, which can further drive hunger and cravings, reinforcing the cycle. Stress and the body's stress response system play an important role. Activation of the hypothalamic pituitary adrenal axis leads to cortisol release, and cortisol can increase appetite and preference for energy-dense foods. Chronic stress can make the brain more sensitive to rewarding stimuli such as palatable food, increasing the likelihood of binge episodes. In some individuals, binge eating becomes a habitual stress response, with the behavior deeply ingrained in neural pathways that link stress cues to eating behaviors. Environmental and learned associations further contribute to the persistence of binge eating disorder. Over time, the brain can become conditioned to associate certain times of day, locations, or emotional states with binge eating. These cues can trigger cravings even without physiological hunger. Because these learned associations are stored in long-term memory and reinforced through repeated behavior, They can be powerful drivers of relapse even after periods of abstinence. Genetic predispositions also play a role in vulnerability to binge eating disorder. Variations in genes related to dopamine and serotonin signaling can influence reward sensitivity, impulsivity, and emotional regulation. Individuals with heightened sensitivity to reward or greater difficulty controlling impulses may be more susceptible to binge eating behaviors. These traits, combined with environmental stressors or food availability, create a higher risk of developing the disorder. The repeated cycle of binge eating can create structural and functional changes in the brain over time. Repeated activation of the reward system in response to food cues strengthens the neural pathways associated with binge behavior, making it more automatic and harder to interrupt. At the same time, repeated emotional reliance on food can weaken alternative coping mechanisms, further embedding food as the primary strategy for emotional regulation. This combination of altered brain circuitry, hormonal dysregulation, and conditioned behavioral patterns makes binge eating disorder a persistent and challenging condition that requires understanding both the neurobiological and psychological drivers that maintain it. Healthy eating relies on the stability and proper functioning of brain circuits that regulate hunger, satiety, reward, and decision-making. These systems work together to ensure that the body receives the necessary nutrients to sustain energy, repair tissues, and maintain cognitive function. One important component is the regulation of meal timing. Regular eating patterns help stabilize hormonal rhythms, including insulin and cortisol, which in turn support balanced energy levels and mood. When meals are spaced consistently, the body can predict when energy will be available. Reducing stress on the metabolic system and avoiding large fluctuations in blood sugar levels that can lead to cravings or fatigue. Balanced macronutrient intake is also essential for healthy eating. Carbohydrates, proteins, and fats each play unique roles in maintaining brain and body health. Carbohydrates, especially complex ones, provide glucose, which is the primary fuel for the brain. Proteins supply amino acids that are used to synthesize neurotransmitters such as dopamine and serotonin, which influence mood, motivation, and appetite regulation. Healthy fats, including omega-3 fatty acids, are critical for maintaining the structural integrity of brain cell membranes and supporting communication between neurons. When these macronutrients are consumed in appropriate proportions, they create a stable internal environment that supports both physical performance and mental clarity. Micronutrients such as vitamins and minerals are equally important. Nutrients like magnesium, zinc, B vitamins, and vitamin D play significant roles in neurotransmitter synthesis, hormone regulation, and energy metabolism. Deficiencies in these micronutrients can impair cognitive function, mood stability, and the ability to regulate appetite. For example, low magnesium levels can increase anxiety and disrupt sleep, both of which can influence eating behavior. Ensuring a varied and nutrient-dense diet helps prevent these deficiencies and supports the optimal functioning of brain and body systems involved in appetite control and decision-making. Mindful eating is another component of healthy eating that has strong effects on the brain. Mindful eating involves paying close attention to the sensory experience of eating, flavors, textures, and satiety cues, while minimizing distractions. This practice activates the insula, the brain region responsible for interoception, which enhances awareness of internal signals like hunger and fullness. Over time, this increased interoceptive awareness helps individuals eat in alignment with their physiological needs rather than external cues or emotional triggers. Mindful eating also engages the prefrontal cortex, strengthening self-regulation and decision-making during meals. The emotional context in which eating takes place is also critical. A healthy relationship with food avoids associating eating with guilt, shame, or anxiety. These emotions can activate the amygdala and stress-related systems, which can disrupt normal appetite regulation and lead to maladaptive eating behaviors. By approaching food without judgment, the brain remains in a calmer state, allowing hunger and satiety cues to guide eating behavior more accurately. This approach reduces the likelihood of cycles of restriction and overconsumption that can develop when shame is attached to eating. Environmental factors also influence healthy eating behaviors. The availability of nutrient-dense foods, the presence of supportive eating environments, and the absence of highly processed, hyper-palatable foods in immediate surroundings all shape daily choices. Environmental cues interact with brain circuits in the reward system, making certain foods more or less appealing based on exposure. Structuring the environment to favor whole, minimally processed foods reduces reliance on constant self-control and instead aligns the environment with long-term health goals. Social context plays an additional role. Shared meals, cultural traditions, and family eating patterns can support consistency in healthy eating. Eating in social settings where balanced meals are the norm reinforces positive habits, while environments that normalize erratic eating or constant indulgence can make healthy eating more challenging. The brain's mirror neuron system and social reinforcement mechanisms make us more likely to adopt the eating patterns we observe in others, which means social environment directly influences personal dietary behavior. Another important element of healthy eating is flexibility. Rigid rules around food can create unnecessary stress, while a flexible approach allows for enjoyment of a wide variety of foods without triggering guilt or anxiety. This flexibility engages the prefrontal cortex in adaptive decision-making, supporting both short-term enjoyment and long-term health. Over time, this adaptability helps prevent the kind of all or nothing thinking that can lead to disordered eating patterns. Healthy eating is sustained by the integration of these biological, psychological, and environmental factors. Stable brain circuits that regulate appetite, reward, and decision-making interact with supportive environments and nutrient-dense diets to maintain both physical and mental health. Consistency, balance, mindfulness, and a positive relationship with food work together to create an eating pattern that supports energy, mood, and resilience over the long term. Removing shame from food choices is essential for breaking disordered patterns and restoring a healthy relationship with eating, because shame activates neural and hormonal systems that interfere with normal appetite regulation and decision-making. Shame is a powerful emotional state that engages the amygdala, a brain region responsible for processing threat and negative emotion. When shame is triggered in the context of eating, the amygdala signals the hypothalamus and the hypothalamic pituitary adrenal axis, leading to the release of cortisol. Elevated cortisol can disrupt hunger and satiety cues, increase cravings for high-calorie comfort foods, and reinforce stress-related eating patterns. This creates a physiological loop in which the very emotions meant to promote restraint actually drive more dysregulated eating behaviors. Shame also interacts with the brain's prefrontal cortex, which is responsible for planning, self-regulation, and rational decision-making. In moments of high emotional intensity, such as feeling guilt or shame about food, the prefrontal cortex's ability to apply logical thinking is diminished. This can lead to impulsive food decisions driven by short-term emotional relief rather than long-term health goals. Over time, repeated cycles of shame and dysregulated eating can weaken the functional connection between the prefrontal cortex and other brain regions involved in emotional regulation, making it harder to interrupt the pattern without intentional intervention. From a psychological perspective, shame tends to create avoidance behaviors. When individuals feel ashamed about their eating choices, they may avoid situations where their food intake could be observed, leading to more isolated eating patterns. This isolation can increase vulnerability to disordered eating behaviors such as binge eating, where episodes take place in private and away from social accountability. Shame also contributes to black and white thinking about food, where certain foods are labeled as entirely good or bad. This binary thinking can make minor deviations from strict rules feel like failures, which may lead to giving up entirely and overeating in response. Social influences amplify the effects of shame. Cultural pressures, diet culture, and media representations of idealized body types contribute to an environment where eating choices are often judged. The brain's social processing systems, including mirror neurons and circuits related to social evaluation, are sensitive to perceived judgment from others. Even the anticipation of judgment can activate stress responses, leading to changes in eating behavior. Over time, repeated exposure to these pressures can normalize the experience of shame around food, making it a default emotional reaction rather than an occasional event. Physiologically, repeated experiences of shame can alter brain chemistry in ways that reinforce disordered patterns. Chronic stress and elevated cortisol can impact neurotransmitter systems such as serotonin and dopamine, which are involved in mood regulation and reward processing. Low serotonin can increase anxiety and impulsivity, while altered dopamine signaling can make high reward foods more appealing as a way to self-soothe. This neurochemical environment makes it harder to approach food neutrally, and instead food becomes tied to emotional highs and lows. Breaking the cycle of shame involves creating an emotional and cognitive environment where food is approached without moral judgment. This allows the brain's regulatory systems to function without interference from stress-related circuits. When food is viewed as a neutral source of nourishment and enjoyment, the amygdala remains calmer, cortisol levels stay more stable, and the prefrontal cortex can guide decision-making more effectively. Over time, repeated experiences of eating without shame can rewire neural pathways to reduce emotional reactivity around food. Developing self-compassion and non-judgmental awareness also plays a role in weakening shame's hold on eating behavior. Engaging the brain's default mode network through reflective practices can help shift perspective from self-criticism to curiosity. This shift supports emotional regulation and allows for greater flexibility in eating decisions. As the emotional charge around certain foods decreases, the brain's reward system becomes less reactive, reducing the intensity of cravings and the likelihood of binge or restrictive episodes triggered by guilt. The social context in which food choices are made can either reinforce or reduce shame. Supportive environments where diverse eating patterns and body types are normalized help reduce the perception of judgment. In these settings, the brain's social circuits are less likely to interpret eating behavior as a potential source of rejection or criticism. This makes it easier to maintain balanced eating habits without the added emotional burden of shame. Removing shame from food choices ultimately supports the stability of brain circuits involved in appetite regulation, decision-making, and emotional balance. By reducing activation of stress-related systems, maintaining stable neurotransmitter levels, and strengthening prefrontal control, individuals are better able to engage in consistent, flexible, and nourishing eating patterns. Over time, this promotes a sustainable and healthy relationship with food that is resilient to external pressures and internal emotional fluctuations. Eating is one of the few behaviors we perform multiple times a day, every day for life. It's wired into our survival circuitry. But when that wiring goes awry, through trauma, stress, or neurobiological changes, eating becomes disordered. Healing is possible. Not through force or judgment, but through understanding the brain, the body, and the powerful feedback systems that govern how we nourish ourselves. And that understanding starts here with knowledge, compassion, and science.